If you think of DNA as a twisted ladder, nitrogenous bases are the rungs. They are shaped like flat rings stacked on top of each other in the middle of the DNA strand. Their function is to spell out the sequences that we call genes. You can think of the bases as letters and the genes they make as words or sentences. There are 4 bases in DNA, and each one is different than the others, making 4 letters. Amazingly, this is enough variety to spell out all the genes in all living things. The related molecule RNA also has 4 bases. 3 are the same as in DNA, and 1 is different. Let's meet the different nitrogenous bases. Adenine, abbreviated A, has a 2-ring structure, so that makes it a purine. When it's in DNA, it pairs up with thymine. When it's in RNA, it pairs up with uracil. Adenine also has other jobs in the cell. It makes up part of the energy molecule ATP and electron carriers FAD and NAD, which are used in cellular respiration. Thymine, abbreviated T, is a pyrimidine, which means it has a 1-ring structure. It's only present in DNA where it pairs with adenine. As we just noted, its cousin uracil does the same job in RNA. Uracil, abbreviated U, is found in RNA. It's a pyrimidine and it bonds with adenine. Think of it as having the same job in RNA that thymine does in DNA. Guanine, abbreviated G, is part of both DNA and RNA, where it bonds with cytosine. Guanine is a purine. Fun fact, it was first discovered in bird feces known as guano, hence the name. Cytosine, abbreviated C, is part of DNA and RNA and bonds with guanine. It has 1 ring, so it's a pyrimidine. So what's in a nitrogenous base? The first clue is in the name. They contain nitrogen, which you can see as the letter N in the pairing diagrams. While carbon always makes 4 bonds to other atoms, nitrogen makes 3 real covalent bonds and has an extra pair of electrons. This pair makes the nitrogen slightly negative so it can be attached to hydrogen and form a weak hydrogen bond. We'll talk more about that in the section on pairing up. As I mentioned earlier, there are 2 types of nitrogenous bases: purines, which are shaped like a double ring, and pyrimidines, which are shaped like a single ring. Having trouble remembering which ones are purines? Think of the phrase, girls are pure, for guanine and adenine, and imagine a girl wearing 2 hoop earrings. Note that a purine with 2 rings always bonds to a pyrimidine with 1 ring, which fits perfectly within DNA. Each base pair is always the same size, 3 rings wide. The rungs are flat, just like the rungs of a real ladder, which help the bases stack without interfering with each other. Nitrogens on one base are attracted to hydrogens on its partner, which we call a hydrogen bond. Think of how static electricity can make a sock stick to your pants when they come out of the dryer. The sock is easy to remove, but it will stick there until you take it off. Hydrogen bonds are like that. They're not a true covalent bond, just an attraction that keeps things together temporarily. Because the hydrogen bonds are weak, enzymes can easily break them when it's time to open up the strand of DNA to make a copy. And because each base always has the same partner, the enzymes know which base to put on the matching strand. For example, if you have a strand with an A, you know that the other strand should have a T in that same spot. Let's review. If you think of DNA as a twisted ladder, nitrogenous bases are the rungs. They are shaped like flat rings. Their function is to be the letters that spell out a sequence. Some, like adenine, also have other roles in the cell. Nitrogenous bases make hydrogen bonds that let them pair up with their partner. In DNA, it's G to C and A to T. In RNA, U takes the place of T. The bonds are strong enough to hold the double DNA strand together, but weak enough that enzymes can easily unzip the strand when it's time to make a copy.